2. You might be wondering what this statistic is referring to. 1 in 2 Australians will be diagnosed with cancer by the age of 85. 1 in 17 will be diagnosed with melanoma, a skin cancer of the melanocytes, by the age of 85. As you might know, Australia has one of the highest rates of skin cancer in the world. Melanoma alone causes 71% of skin cancer deaths. However, it's not all bad news. At stage 4, the survival rate is 26%. But at stage 1, it's 99%. So we know the earlier we can detect melanoma, the better the outcomes for patients and the healthcare system. The current gold standard for melanoma detection is biopsies. This is where either the entire mole or a section of the mole is cut out of the skin. Obviously, this is quite invasive and can cause some significant scarring, and the results can take a while to come back. There's also a lack of point-of-care testing. So this is a type of testing that is done near the patient without needing an expert at the lab to do the test. Some common examples include COVID-19 rats and pregnancy tests. So this is where we come in. We are SynBioM, and we hope to provide a solution to the lack of point-of-care tests. We are Monash's first team to take part, and this is our student team here. We come from a range of different backgrounds, including computer science, engineering, and biomed. So, on to the project, the Monash Melanoma Multiplex. We've been looking at detecting melanoma-specific microRNAs using a CRISPR-Cas13 bioassay. So microRNAs are small, single-stranded RNAs that are important in the regulation of gene expression. In melanoma, they're often involved in tumour development. We'll go through the exact mechanisms of this bioassay later. The idea is that we could eventually develop this bioassay into a point-of-care test, but this takes many years to develop. What we've been looking at is whether it's possible to actually detect the microRNAs, and then whether we can accurately quantify the concentrations. So based on the literature we read, there were 3 main takeaways. So first off, there are 7 microRNAs that are either upregulated or downregulated in melanoma. We were able to order 6 of these through RDT. This panel is known as the MAL-MIR7 panel. 2, the cleavage ability of CRISPR-Cas13 allows for high accuracy and rapid quantification of specific microRNA concentrations. This is specifically quite exciting because methods like detector and Sherlock are often only qualitative, and so getting quantities would be really novel. And then lastly, the concentrations of the microRNAs can be quantified using fluorescent probes. So essentially, when microRNAs are detected by CRISPR-Cas13, the solution will fluoresce.
So, how does this work? Well, that's a great question. Well, it all begins with 4 simple components. We have 1, Cas13, 2, the guide RNAs, 3, the target microRNAs, and 4, the fluorescent probes. We take these components and we create 2 simple mixtures. One mixture is Cas13 and the guide RNA, essentially giving Cas13 the instructions on which microRNA out of our selected 7 to actually target. The other mixture is the microRNA and the fluorescent probes. Currently, the fluorescent probes are inactive due to a quencher molecule being attacked, halting the probe from actually fluorescing. Next, we actually take both of these mixtures and combine them. The instructed Cas13 then finds, binds, and cleaves the target microRNA. This is a process called cis cleavage, which, much like Cas9 you may have heard of, is able to target and cleave specific sites of DNA. However, this binding of Cas13 activates a unique nature of it, a different process called transcleavage, whereby the activated Cas13 starts to cleave any surrounding RNA in the mixture. This is sort of like collateral damage, and it especially likes to target uracil-rich RNA. So the uracil-rich fluorescent probes are now split in half, separating the quencher molecule from the fluorescent molecule, allowing the probe to fluoresce unimpeded and to be detected by our plate reader. So we can think about it, the more target microRNA there is, the more Cas13 is activated, the more transcleavage there is, the more fluorescence will occur. So therefore, we're actually able to correlate total fluorescence to target microRNA concentration in these mixtures. Our our recent experiments have all been about creating calibration curves. So getting ideas, trying to see how much fluorescence will occur for different microRNAs at different concentrations. So that later, when we wish to determine unknown concentration of microRNAs in samples, we can compare the fluorescence, or we can conduct our Monash multiplex, compare the fluorescence to the calibration curves and get a concentration. The calibration curves are determined through conducting what's called triple replicas. So essentially running the experiment 3 times concurrently and averaging the results from our plate reader. Our optimized experimental procedure was derived with help from the not laboratory part of Monash Biomedical Discovery Institute. And here's some pictures of us conducting our experiments. I'm going to hand it over to.
Thank you, Andrew. Aren't you glad you don't actually have melanoma yet? So, hi everyone. Sorry, that was so funny. Stop. I'm Annabelle, and I'll be discussing our results with you. And essentially, we were able to get data from microRNA fluorescence at different concentrations along with our positive control. And initially, we tried to mess with the parameters of our plate reader, where we tried to change the working concentrations, temperatures, pipetting instruments, and our gain function so that we can get appropriate fluorescent detection. And thus far, we've been able to we've been able to investigate the microRNA strands 5095P, 165P, and 47315P. And you can see here on the slide that our positive control for the 10 nanomolar concentration, although it's the highest concentration that we've used, the fluorescence is lower, and even though it could be because of gain function or calibration of our plate reader, we hypothesized that this is because the concentrations are too high for the fluorescence to be properly detected. And this is also seen in our reference by our partner, the NOT lab. And so from now on, I'll just be talking about concentrations from 10 femtomolar to 10 picomolar, because this is sufficient for us to detect fluorescence in the microRNAs for melanoma. So, in our triplicates, we can see that the primary factors affecting the speed of fluorescence detection is the gain function setting of our plate readers. Changing this allows us to detect fluorescence in more accuracy and efficiency. And hence you can see that there's an increase in the slope inclination between these two graphs. And there is also a greater error between these two. That's because we use a multi-channel pipette between these for more efficiency, but that will also cause greater inaccuracies because of the small sample size used. And there's also another factor, and that would be temperature. You can see that on the right graph, the slopes are not very easily differentiated between the concentrations. That's because we use the temperature of 22 to 24 degrees Celsius. But over here, on the right, you can see that using a temperature of 37 degrees Celsius is a more optimal incubation temperature that mimics the condition of our Cas13 functionality more accurately. So we managed to get sound results for our microRNA 165P. Alas, as we are in the initial phases of our experimentation, there are multiple limitations that we have yet to conquer. Although we have tried to maintain a lot of consistency, there will be factors like the varieties between time taken to activate the samples and also for us to detect the fluorescence. And our microRNA concentrations used are also unknown, and they may vary. Additional limitations would be contamination. As you can see here, we can't use these results at all. But there's also other things like fluorescent noise, pipetting variability, and the sample sticking to the plate wells. So there's like a homogeneous sample mixture. And this could be easily resolved through proper practice and sterilization, replicating more experiments, and also optimizing our methods. Thank you.
Thank you very much for that. Okay, so as Annabelle just walked you through, our key finding here is that out of the 3 different panel members for MEL MI7, we were able to test 3, and we found that all of them detected signal with Cas13a. And I want to emphasize that this isn't necessarily always the case. So a lot of the time, your RNA targets will be fairly long. And so a lot of the time, researchers will be designing multiple guide RNAs for each microRNA target, for each RNA target. And then they'll test these different guides to see which ones give a large fluorescent signal over time and which ones don't deviate at all. So I've included this figure here, which is from Fazuni and colleagues from 2021 paper. I believe they looked at the COVID's N gene, and they looked at 12 different guide RNAs. And you can see in the second figure over there, you get a very steady and large increase in fluorescence over time. So this is a successful guide RNA. But if you take a look at the first one and the 12th one, you can see there's barely any difference from any sort of basal control level increase in fluorescence. So, something to point out is that in a clinical setting, doctors and patients need not wait around for 2 hours to see these steady rises in fluorescence as we did during our lab work. Instead, if you look at our error bars, in this case, they're extremely small for the positive control, so you won't actually see them. Oh, you do see them a little bit, all right then. But you can see they differentiate, these lines differentiate before 30 minutes of testing time. And this indicates that we could create a point of care test that only lasts 30 minutes in comparison to other techniques like a qPCR, where you might need to leave it in for a little bit longer than that to see a change. And these kind of characteristics are really suitable for a point of care test where you don't want to be waiting around for too long. However, our results are not without limitations. Annabelle did discuss a few of them. Another one I want to point out is that if you look at the very start of these graphs, you'll see that we haven't quite captured the initial rise up to those higher levels of fluorescence. And this is because for a lot of our experiments, we didn't have access to the right multi-channel pipette. So with the plate reader having 48 different wells, we need to quickly start the reactions and get it into the plate reader, but we had to individually pipette these instead. And this can take a few minutes. So you can imagine that by the time you're at the last well, the first well's reaction has already started for a few minutes. And that explains why some of the lines begin a little high there. This also, for our purposes, means that it can skew comparisons between concentrations a little bit, because these reactions can be a little asynchronized as well. So although we're very interested in the whole MEL MIR7 panel, currently we've only been able to test 3 out of the 7, and we found success for all 3 of those. So moving forward, we'd like to test the other 4 members in the panel, and then following all of this, we'd like to look at real melanoma serum samples, and possibly also saliva samples, and see if we can apply the miRNA detection methods with Cas13 to these biological samples directly. Something else I also want to mention is that this RNA work is extremely precise. So some of you have also worked with microRNAs, and you'd be very familiar that you need to use these very special cleaning solutions. There are all these different sort of techniques surrounding that, and also RNAs being extremely sensitive. If you breathe on it, it's probably going to break down. So with that note, I think one of the possible sources of innovation with producing this kind of assay on scale is to integrate automation alongside this and give clinicians access to pre-prepared reagents. So with our lab work, we often have to pipette around multiple tubes and add all these different components together. But if clinicians were able to receive tubes that already had the main components in, and then they can use automation to simply mix these components. And there are other innovations coming out of different researchers as well, looking at using mobile phone microscopy to detect fluorescence. So they simply utilize a mobile phone camera and can efficiently detect fluorescence without the need for even a plate reader. So that's stuff that's really exciting to us as well. And so in saying that, I want to extend our gratitude to those who have helped guide us along the way, in particular our PI Simon Corrie, and all of our advisors such as Dr. Andrew Perry, Dr. Gavin Knott, Ankit Dodler, Prasant Ramesh, and Nathan Chai. And furthermore, I'd also like to thank the AusSynBio organizers for coordinating this whole event, and also the sponsors listed on screen for making the time to meet with us, being excited about these ideas, and also funding some of our work. Thank you very much.
you.